Welcome everyone. I, Ritika Manglik, volunteer of AFRS, Applied Forensic Research Sciences, is going to present on the topic of DNA fingerprinting today. Table of contents. First, characteristics of DNA. Second, DNA fingerprinting in forensic science. Third, origin, history of DNA fingerprinting. Fourth, history of DNA profiling, DNA in civil cases. Fifth, how DNA fingerprinting works. Sixth, application of DNA fingerprinting. Seventh, case studies. And eighth, conclusion. Characteristics of DNA. DNA, which stands for deoxyribonucleic nucleic acid, is the hereditary material in almost all living organisms. It carries the instructions for building, maintaining, and regulating the functions of cells and organisms. DNA composed of two long chains of nucleotides twisted into a double helix structure. DNA fingerprinting is an essential tool in forensic science which does not precisely determine the suspect's identity but helps narrow it down, like which suspect's DNA closely matches the sample from the crime scene. DNA double helix. It is related to genomics, which is used to describe the physical structures of the DNA. Each DNA molecule is made up of two linked strands that wind around each other to resemble a twisted ladder in a helix like shape. The backbone of DNA is nucleoside, which is made up of alternating sugar, deoxyribose, and phosphate groups. Each sugar is attached to one of the four bases: adenine, cytosine, guanine, or thymine. The two of the strands are connected by hydrogen bonds between the bases of adenine bonds with thymine and the cytosine bond with guanine. DNA fingerprinting in forensic science. It is a technique used to identify individuals based on unique patterns in their DNA. It is widely used in criminal investigations, paternity testing, and identifying victims. First, DNA extraction. DNA is collected from the biological samples like blood, hair, or skin cells. Second, amplification PCR. Specific regions of the DNA, particularly short tandem repeats (STRs), are amplified using polymerase chain reaction PCR. Third, analysis. The number of repeats in the STR region varies between individuals, and this variation is used to create a unique DNA profile. Fourth, comparison. The DNA profile obtained from the crime scene sample is compared to those of suspects or databases. Matching profiles can help identify individuals or exclude suspects. This technique is highly accurate because the probability of unrelated people having identical STR profiles is extremely low. Origin of DNA fingerprinting. The origin of DNA fingerprinting dates back to the 1980s, with its development credited to British scientist Alec Jeffreys. In 1984, Alec Jeffreys, a geneticist at the University of Leicester, made a groundbreaking discovery while studying satellite DNA, regions of DNA that are highly variable among individuals. These regions contain repeating sequence of nucleotides which vary in the number of repeats from person to person. Jeffries noticed that these variations were unique to individuals, much like a fingerprint. He realized that by analyzing these specific regions of DNA, one could potentially identify people with great precision. He called this technique DNA fingerprinting. History of DNA fingerprinting. The Linda Mann and Don Ashwood case is the significant example of how DNA fingerprinting was used in criminal justice to solve a double murder case in the UK during the 1980s and the 1990s. The case also marked one of the first instances in which DNA evidence was used to convict, suspect, and exonerate others. In 1980s, two young girls, Linda Mann and Dawn Ashwood, were sexually assaulted and murdered in the Leicestershire area of England. Their deaths remained unsolved for several years, and the case became a mystery that the police struggled to crack. In 1986, Alec Jeffreys, the British geneticist who had pioneered the DNA fingerprint technique, was approached to help in solving the case. This was a crucial turning point in the investigation. Here, DNA evidence was collected from both the crime scenes, specifically from semen found on both victims. Using Jeffrey's newly developed DNA fingerprinting method, investigators compared the DNA samples from the two murders. They found that the semen samples from both victims came from the same man. The DNA analysis provided the police with a significant lead. They had a genetic profile of the perpetrator. The big breakthrough came when the DNA profile was compared to the local male population. Colin Pitchfork, a local man with a history of sexual offenses, was identified as a potential suspect. Pitchfork had not yet been directly implicated in the murders. Pitchfork, who had been living in the same area at the time of the murders, was not initially a suspect, but DNA profiling allowed the police to focus on him. Pitchfork's DNA was finally tested and matched the DNA found at the both crime scenes. Then he was arrested in 1987 after his DNA was confirmed to match the evidence collected from the victims. The table outlines significant milestones in forensic DNA science and its applications from 1985 to 1992. In 1985, Alec Jeffreys develops multilocus RFLP restriction fragment length polymorphism probes, marking the advent of modern DNA fingerprinting. In 1986, DNA testing becomes publicly accessible in the United States with companies like Cellmark and LifeCodes. In 1988, the FBI initiates the DNA casework using single locus RFLP probes. In 1989, the Technical Working Group on DNA Analysis Methods is established to address DNA quality standards. Additionally, the New York versus Castro case raises questions about the quality assurance of DNA laboratories. In 1990, concerns emerge about the statistical methods used with RFLP techniques. PCR polymerase chain reaction methods are introduced, starting with GQ alpha typing. In 1991, fluorescent STR short tandem repeat markers first described and the Chilex extraction method is introduced for DNA sample preparation. In 1992, the first NRC National Research Council report on DNA testing is published. The FCI begins casework using PCR DQ alpha techniques. This timeline highlights development and adoption of key technologies and methodologies in forensic DNA analysis. History of DNA profiling. DNA profiling, also known as DNA fingerprinting, is a forensic technique used to identify individuals based on their unique DNA. In 1984, Sir Alec Jeffreys, a British geneticist, develops DNA fingerprinting using restriction fragment length polymorphism analysis. This revolutionizes forensic science by providing a reliable method for individual identification. In 1985, the technique is first used in the criminal case in the UK to solve a double murder, exonerating one suspect and convicting another. 1986, DNA testing becomes commercially available through companies like Cellmark and LifeCodes, particularly in the United States. In 1988, the FBI begins using DNA profiling in casework, starting with single locus RFLP probes. In 1990s, the transition from RFLP to PCR methods enables faster and more accessible DNA analysis. STR markers are introduced, making DNA profiling more precise. In 1995, the UK's National DNA Database is established, becoming the world's first DNA database for convicted offenders. In 2000s, YSTRs and mitochondrial DNA analysis are developed, expanding profiling capabilities. DNA databases grow significantly worldwide. At present, DNA profiling is widely used in criminal investigation, paternity testing, and identifying remains in mass disasters or missing persons case. This technology has transformed forensic science, making it cornerstone of modern criminal justice system. DNA in civil cases highlights the role of DNA testing in civil legal matters. Usage in civil disputes. DNA tests are primarily used in civil cases to resolve paternity issues. These are often central to disputes in matters of divorce, maintenance, inheritance, and succession. Legal provision under Section 112 Section 112 of the Indian Evidence Act assumes the legitimacy of a child born during wedlock, establishing a presumption that the husband is the father. The only way to challenge this presumption is to prove non access of the husband during the time of conception. DNA tests are sometimes required to settle disputes when the legitimacy of the child is questioned. Judicial considerations. Initially, courts debated whether DNA tests could be ordered in such cases, especially when it could infringe on privacy rights or contradict legal presumption. Over time, courts have increasingly relied on DNA evidence to ensure justice and resolve doubts. This topic emphasizes the balance courts must maintain between legal presumptions, privacy rights, and the scientific accuracy of DNA testing in civil cases. How DNA fingerprinting works. DNA fingerprinting, also known as DNA profiling, is a technique used to identify individuals based on their unique DNA sequence. First is the sample collection. Biological samples like blood, saliva, hair, skin cells, or semen are collected. These samples contain that individual's DNA. Second step, DNA extraction. DNA is isolated from the cells in the sample using chemical or the mechanical methods. Third, DNA fragmentation. The extracted DNA is cut into smaller fragments using restriction enzymes that target specific sequences. Amplification, if needed, in cases of limited DNA, the polymerase chain reaction PCR is used to amplify specific DNA regions. Fifth, separation by gel electrophoresis. DNA fragments are separated based on their size by passing them through a gel under an electric field. Smaller fragments move faster than larger ones. 6. Transfer to a membrane, southern blotting. The separated DNA fragments are transferred to a nylon or nitrocellulose membrane for further analysis. 7. Hybridization with DNA probes. The membrane is exposed to radioactive or fluorescent DNA probes that bind to specific DNA sequences, highlighting the targeted regions. 8. Visualization. The pattern of DNA bands is visualized using X ray film or fluorescence imaging. This pattern is unique to each individual except for identical twins. Comparison. The DNA fingerprint from the sample is compared with other samples to establish identity or relationships. DNA fingerprinting is highly accurate and widely used for identification purposes in various fields. Applications of DNA fingerprinting. DNA fingerprinting has diverse applications across various fields, including forensics, medicines, and agriculture. Some of them are in forensic science. It is helpful in identifying suspects by comparing DNA from crime scene evidence to potential suspects. Recognizing remains in mass disasters, accidents, or criminal cases. Proving wrongful convictions by reanalyzing old evidences. In paternity and family disputes, determining biological relationships between a child and alleged parents, verifying family relationships in immigration applications, resolving disputes over property and inheritance by establishing familial ties. In medical and genetic research, understanding genetic predisposition to the diseases, matching donors and recipients for organ or tissue transplantation, studying genetic diversity and hereditary disorders. In legal and civil matters, resolving cases of identity fraud or establishing rightful citizenship, providing evidence in family law cases involving parentage disputes. DNA fingerprinting's reliability and precision make it a valuable tool in these fields, offering solutions to complex biological and legal problems. Case studies. The key criminal case studies categorized into international and Indian contexts, emphasizing significant legal milestones and landmark judgments. Internationally, it highlights the Boston Strangler case and specifically mentions the Colin Pitchfork case, which marked the first conviction using DNA fingerprinting in the UK. Evidence derived from blood samples conclusively linked Colin Pitchfork to the crime scene, setting a precedent for forensic science in criminal investigations. In India, the Gudia rape case and the double murder case, both of which drew significant public and legal attention. Additionally, Two highlighted cases: the Priyadarshini Mattu case, known for its prolonged legal battle and eventual justice, and the Nirbhaya case, which became a symbol for the fight against sexual violence and led to significant legal reforms in India. Conclusion: DNA fingerprinting has revolutionized forensic science and personal identification, providing a reliable method for solving crimes, establishing paternity, and identifying remains. The technique analyzes unique patterns in persons' DNA, particularly in regions that are highly variable between individuals. While DNA fingerprint is highly effective, it raises concerns related to privacy, consent, and the potential for misuse in areas such as surveillance and data storage. Legal frameworks must continue to evolve to address these challenges while preserving its forensic integrity. Ongoing research aims to further refine the technique, improve the speed and cost of analysis, and explore applications beyond forensics, such as in personalized medicine and ancestry testing. DNA fingerprinting remains a cornerstone of modern forensic science, providing irrefutable evidence in many criminal and legal cases. However, its continued use requires responsible handling and ethical considerations. Thank you.